Hey guys, tonight we're venturing into the hidden landscapes of our own bodies as we uncover how sperm is born from countless cellular dances. We're peering beyond what meets the eye to witness the elegant processes that spark new life at the microscopic level. If you love these deep dives into human anatomy, a like and subscribe helps us keep exploring, and let us know where you're watching from and what time it is for you. Now get comfortable, clear your mind, and prepare to drift through the serene rhythms of cell division and maturation. Let's start in the deep, quiet center of the brain. Here within the soft, protected tissue of the hypothalamus, a silent, rhythmic pulse orchestrates a process that will unfold far below. Imagine a slow, steady clock, not of ticking gears, but of hormonal release, a gentle ebb and flow that has persisted almost unnoticed since puberty. You can almost sense it as a faint, rhythmic warmth, a deep and primal cadence that connects your mind to the very origins of life. This is where the initial command is given, a subtle but insistent whisper that travels on the unseen currents of your internal ocean. The hypothalamus releases a molecule, a tiny key known as gonadotropin-releasing hormone. It is a messenger with a single profound purpose, dispatched into the bloodstream in carefully measured bursts, each one a quiet affirmation of potential. This microscopic messenger does not travel far on its initial voyage. It navigates a specialized miniature network of blood vessels, a private channel leading directly to the pituitary gland, nestled at the base of your brain. Think of it as a secret passage, ensuring the message is delivered with precision and without dilution. As the gonadotropin-releasing hormone arrives, it finds its destination on the surface of pituitary cells, fitting perfectly into receptors like a key into a lock. This gentle docking initiates the next phase of the cascade, another silent instruction passed down the chain of command. The pituitary gland, now activated, responds by releasing two more powerful messengers into the wider circulation of your body. These are luteinizing hormone and follicle-stimulating hormone, and their journey will be much longer, a systemic voyage to the core of creation. You can almost perceive the whisper of your own blood coursing through its vast network of capillaries, a river of life carrying these hormonal signals. Feel the gentle, rhythmic pressure of your own pulse, each beat a propulsion system for these microscopic envoys. Luteinizing hormone and follicle-stimulating hormone are swept along in this current, traveling down from the seat of consciousness, past the heart, through the torso, on a mission to awaken a deeper potential. Their destination is a pair of organs held outside the main cavity of the body, a place where the temperature is kept slightly cooler, a crucial environmental detail for the delicate process that is about to begin. The journey is a testament to the body's intricate communication system, a conversation conducted not in words but in molecules, a dialogue between brain and body that ensures the continuation of life. The testes, suspended in the scrotum, await this chemical signal. Imagine them as two secluded laboratories, cradled in soft tissue, where the temperature is maintained at a precise 2 to 3 degrees Celsius below the core body temperature. This subtle coolness is not an accident. It is a fundamental requirement for the viability of the cells you are about to witness. As the luteinizing hormone and follicle-stimulating hormone arrive, carried by the testicular artery, they permeate the dense tissue, their presence a catalyst for profound transformation. The luteinizing hormone seeks out specific cells nestled between the primary structures of the testes, the Leydig cells. Its message is simple and direct. Begin production of testosterone. This powerful androgenic hormone is the primary fuel for spermatogenesis, the very essence of masculine cellular identity. The follicle-stimulating hormone, meanwhile, has a different but equally vital target. It journeys to the wall of incredibly fine coiled tubes that make up the bulk of the testicular mass. These are the seminiferous tubules, and if you could unravel and lay them end to end, they would stretch for an astonishing 700 meters, a vast, microscopic network packed into a remarkably small space. Here, the follicle-stimulating hormone communicates with the Sertoli cells, large, nurturing cells that form the very architecture of these tubules. The message it delivers is one of preparation and support, nourish and protect the developing germ cells. Without this constant support from the Sertoli cells, the entire process would falter and fail. Together, The surge of testosterone from within the testes and the nurturing signal from the brain create the perfect hormonal environment, a fertile ground for the genesis of new life. This intricate hormonal dance is the unseen prelude to the main event. It is a constant humming background process, a state of readiness maintained by the body's deep wisdom. Feel the subtle warmth of this region, a warmth generated by the constant influx of blood and the ceaseless metabolic activity occurring within. This is not a hot, aggressive process, but a gentle, sustained warmth, like an incubator carefully calibrated to nurture the most delicate of beginnings. The air around you might feel still, but within this microscopic landscape, there is a constant, purposeful flow. Blood brings oxygen and nutrients, hormones deliver their instructions, and a deep cellular rhythm begins to stir, awakened by these chemical whispers from the brain. The very structure of the testes is a marvel of biological engineering, designed for maximum output and protection. Each testes is encased in a tough, fibrous outer layer called the tunica albuginea, a protective white coat that provides structural integrity. From this outer layer, thin walls or septa extend inward, dividing the testes into about 250 to 300 small compartments, or lobules. It is within each of these lobules that the magic truly begins, for each one contains 1 to 4 of the tightly coiled seminiferous tubules. This compartmentalization ensures that the process is organized and contained, a vast production line housed within a secure and stable environment. Imagine these tubules not as simple pipes, but as complex, living structures, their walls lined with the very cells that will give rise to sperm. The testosterone now begins to saturate the testicular tissue, its concentration here hundreds of times higher than in the general bloodstream. This incredibly rich local environment is the key that turns the engine of sperm production. It stimulates the Sertoli cells to produce androgen-binding protein, a molecule that cleverly traps testosterone within the seminiferous tubules, ensuring a constant high-level supply directly where it is needed most. This localized concentration is a beautiful example of the body's efficiency, creating a specific microenvironment perfectly tailored to a single vital task. The stage is now set, the hormonal signals have been sent and received, the environment is primed, and the raw materials are in place. The journey into the heart of the seminiferous tubule, into the very genesis of a sperm cell, is about to begin. The process is ceaseless, a continuous production that from this moment forward will generate over 1,000 sperm with every beat of your heart. This initial phase, governed by the silent language of hormones, establishes the foundation for everything that is to follow. It is a testament to the profound connection between the brain and the reproductive system, a link that ensures the perpetuation of the species is guided by a central command center. The gentle, rhythmic release of gonadotropin-releasing hormone is the pacemaker of this entire system, a steady, reliable pulse that ensures the production line never truly stops. As you settle deeper into this narrative, allow your own breath to mimic this slow, steady rhythm, a conscious mirroring of the unconscious, life-affirming processes unfolding within. You are a universe of intricate, coordinated actions, and this is but one story, one deeply fundamental process, unfolding in the quiet, protected spaces of your own anatomy. The hormonal tide is high, the environment is rich and waiting, and the first cellular actors in this grand play are about to take the stage, responding to the ancient chemical call to create. The sensation is one of profound, internal readiness. It is the quiet hum of a perfectly tuned machine, operating in the background of your awareness, a testament to the autonomous, self-sustaining nature of life. Every signal has been meticulously delivered, every preparation has been made. The Leydig cells are dutifully producing the vital testosterone, and the Sertoli cells, bathed in follicle-stimulating hormone, are prepared to nurture and guide. The very air you breathe seems to carry a sense of potential, a quiet anticipation of the cellular marvels to come. This is the moment before the first division, the pause before the symphony of creation begins in earnest. It is a moment of pure potential, held in the warm, secure cradle of the testes, ready to unfold into a story of microscopic transformation and immense biological significance. The journey is just beginning, and the path leads inward, into the heart of the tubule, where life's blueprint is read and replicated with breathtaking precision. We now journey deeper, past the Leydig cells bathing in their hormonal glow, and move towards the boundary of the seminiferous tubules themselves. Imagine approaching a vast coiled structure, a living filament hundreds of meters long, compressed into a space no larger than a small plum. The wall of this tubule is a complex living barrier, not merely a passive container, but an active participant in the creation of life. As you draw closer, you can perceive its texture, not as a smooth, uniform surface, but as a landscape of interlocking cells, a mosaic of life forming a community with a shared purpose. The tubule is surrounded by a thin layer of contractile cells, myoid cells, whose slow rhythmic contractions help to gently move the developing sperm and fluid along the length of the tube, a first subtle push on their long journey. It is a peristaltic whisper, a gentle muscular pulse that aids the inexorable forward flow. At the very base of this tubular wall, resting on a fine membrane, are the spermatogonial stem cells. These are the foundational cells, the very origin point of every sperm cell that will ever be created. They are nestled here in a special compartment, a protected niche that keeps them safe from the body's own immune system. This protection is crucial, for as these cells begin to change and develop, they will start to display proteins on their surface that the body would recognize as foreign, as not self. To prevent an autoimmune attack, the architecture of the tubule creates a formidable defense, the blood testis barrier. This barrier is formed by the tight embrace of the large Sertoli cells, which are joined together by incredibly dense connections, sealing off the inner sanctum of the tubule from the direct reach of the bloodstream and its immune patrols. The Sertoli cells are the true architects and guardians of this microscopic world. Picture them as tall, columnar cells, standing shoulder to shoulder, their bodies stretching from the outer edge of the tubule, all the way to the central channel, the lumen. They are immense in comparison to the developing germ cells, which they envelop and nurture throughout their entire transformation. You can imagine these Sertoli cells as great, silent sentinels, their cytoplasm rich with nutrients, their membranes studded with receptors, constantly sensing and responding to the needs of the smaller cells they protect. They are the gatekeepers, the nurses, and the structural support of this entire process, creating a perfectly controlled microenvironment where spermatogenesis can unfold in safety and with precision. The follicle-stimulating hormone we spoke of earlier binds to these Sertoli cells, instructing them to facilitate this entire grand production. Within the protective embrace of these Sertoli cells, in the basal compartment outside the blood testis barrier, the spermatogonial stem cells lie in a state of perpetual renewal. With each pulse of testosterone, a cohort of these stem cells is stimulated to divide. This division is a fundamental process of life, known as mitosis. It is a division of replication, creating two identical daughter cells from one parent cell. One of these daughter cells will remain as a stem cell, ensuring the reservoir of potential is never depleted. This is a quiet promise of perpetuity, a cellular mechanism that guarantees this process can continue for decades. The other daughter cell, the type B spermatogonium, is committed to a different fate. It is destined to cross the barrier and begin the journey of becoming a sperm. Feel the gentle rhythm of this constant underlying division. It is not a violent or explosive process, but a slow, deliberate duplication, a quiet hum of cellular activity that forms the baseline of sperm production. For every sperm cell that begins its journey, another stem cell is left behind, a testament to the body's foresight and its commitment to sustainability. This ensures that the production line can run continuously from puberty well into old age, a ceaseless generation of life's potential. The sheer number is staggering. With each cycle, millions of cells are designated for development, a microscopic legion preparing to embark on an incredible transformation. This initial stage, the proliferation of spermatogonia, is the foundation upon which the entire structure of sperm creation is built, a constant replenishment of the raw materials needed for this vital task. Now, witness a moment of profound transition. The committed type B spermatogonium, having divided a few more times to create a small, connected clone of cells, must now move from the basal compartment into the protected adluminal compartment. To do this, it must pass through the blood testis barrier, This is not a simple crossing. The tight junctions between the Sertoli cells must transiently loosen, allowing the developing germ cells to slip through before sealing up again behind them. Imagine a secret door opening just long enough for the initiates to pass into the inner sanctum, a carefully orchestrated event that maintains the integrity of the sanctuary. Once inside, the cells are completely dependent on the Sertoli cells for their survival. They are isolated from the blood, so the Sertoli cells must provide them with all necessary nutrients, oxygen, and signaling molecules, while also diligently removing their waste products. Once safely across the barrier, the spermatogonia undergo their final mitotic division and transform into primary spermatocytes. These are the largest germ cells you will find within the tubule, their nuclei swollen with the genetic material they are preparing to process. They are now officially embarked on the path of meiosis, the specialized type of cell division that will reduce their genetic content by half. The primary spermatocyte contains the full complement of 46 chromosomes, organized into 23 pairs, just like most other cells in the body. However, a sperm cell, destined to fuse with an egg, can only carry half of this information. The next stage of the journey will be a delicate and breathtakingly complex process of genetic sorting and division, ensuring that each sperm receives a unique and correct set of 23 chromosomes. You can sense the change in the environment as you cross this cellular threshold. The adluminal compartment, inside the barrier, has a unique biochemical composition, a carefully crafted soup of molecules secreted by the Sertoli cells. It is a world unto itself, optimized for the intricate dance of chromosomes that is about to occur. The primary spermatocytes are now suspended in this nurturing medium, held in the embrace of the Sertoli cell cytoplasm, slowly being pushed further and further from the outer wall and closer to the central lumen of the tubule as new generations of cells develop behind them. This is a slow, inexorable conveyor belt of life, a physical progression through the tubule that mirrors the developmental progression of the cells themselves. The quiet, rhythmic contractions of the myoid cells on the exterior of the tubule assist in this gentle, directional flow. The scale of this operation is almost beyond comprehension. At any given moment, within the 700 meters of these tubules, you would find waves of cells at every conceivable stage of development. The process is not synchronized throughout the entire length. Rather, it occurs in waves of initiation that spiral down the tubule. This means that at any cross-section of the tubule, you would see several generations of cells layered from the outside in. The spermatogonia at the base, the primary spermatocytes, the secondary spermatocytes, the spermatids, and finally, the mature sperm in the center. This wave-like pattern ensures that the production of mature sperm is not a boom and bust cycle, but a continuous, uninterrupted release, guaranteeing a constant supply is always ready. It is a system of staggering efficiency and elegance. The primary spermatocytes are now poised for the most dramatic part of their transformation. They have grown, they have been protected, and they have been positioned for the next great leap. The journey so far has been one of preparation and positioning, of hormonal whispers and cellular embraces. Now the internal machinery of the cell itself will take center stage, performing a genetic ballet that has been perfected over millions of years of evolution. The quiet warmth of the testicular tissue, the gentle pulse of the surrounding capillaries, the supportive presence of the Sertoli cells, all of these sensory details create the backdrop for the profound event of meiosis. It is here, in this protected, nurturing space, that the genetic blueprint of the next generation will be shuffled and dealt, a process of infinite possibility and breathtaking precision. Within the nurturing cradle of the Sertoli cell, the primary spermatocyte now commits to the profound journey of meiosis. This is not a simple division, it is a meticulous, two-act play of genetic segregation that lies at the heart of sexual reproduction. You can imagine it as a process of deep cellular introspection, where the cell takes stock of its genetic inheritance and prepares to divide it with perfect fairness. The first act, meiosis 1, is where the true magic of genetic recombination happens. It begins as the 46 chromosomes, arranged in 23 pairs, undergo a process called synapsis. The 2 chromosomes in each pair, one inherited from the mother and one from the father, find each other within the crowded nucleus and align with breathtaking precision. forming a structure called a bivalent. Feel the quiet hum of this intricate molecular choreography. It is a dance of homologous pairs, a recognition of shared identity across a parental divide. As these chromosome pairs lie side by side, an even more remarkable event occurs, crossing over. Segments of DNA are exchanged between the maternal and paternal chromosomes, like two friends swapping stories, creating entirely new combinations of genes on each chromosome. This is the primary engine of genetic diversity, the mechanism that ensures each sperm cell is unique, a one-of-a-kind combination of the genetic heritage that created it. Imagine tiny molecular scissors snipping the DNA strands, and then rejoining them in a new configuration, a seamless edit that shuffles the genetic deck with profound implications. Without this step, you and your siblings would be genetically identical, save for the random chance of which chromosome from each pair you receive. This process of crossing over is a moment of pure creative potential. It is a quiet, internal explosion of novelty, generating variations that have allowed our species to adapt and thrive for eons. The primary spermatocyte invests a significant amount of time in this phase, known as prophase 1, as if it understands the gravity of the task it is performing. It can last for several days, a long contemplative period of genetic craftsmanship. The Sertoli cells diligently attend to the spermatocyte during this vulnerable time, providing a stable and supportive environment, ensuring that this delicate process of recombination proceeds without error. The sheer precision required is astonishing. A single mistake in this exchange could lead to a sperm that is not viable. Yet, it happens with remarkable fidelity, billions of times over. Once the genetic shuffling is complete, the chromosome pairs, now intertwined and forever altered, align at the center of the cell. Spindle fibers, microscopic protein filaments, reach out from opposite poles of the cell and attach to each pair. You can almost sense the tension building, a gentle, insistent pull from either side. The purpose of meiosis, one, is to separate these homologous pairs, not to split the individual chromosomes, as in mitosis. The spindle fibers slowly contract, pulling one entire chromosome from each pair to opposite ends of the cell. One pole receives the maternal chromosome, now carrying some paternal genes, and the other receives the paternal chromosome, now carrying some maternal genes, for each of the 23 pairs. The result is two new cells, each containing 23 chromosomes, but with each chromosome still consisting of two sister chromatids. The primary spermatocyte then divides down the middle, a process called cytokinesis, pinching off to form two smaller, distinct cells. These are the secondary spermatocytes. They are transient, ephemeral figures in this grand narrative, existing for only a few hours. Their creation marks the successful completion of the first meiotic division, the reductional division. The cell has successfully halved its chromosome number, from 46 to 23. However, the genetic story is not yet complete. Each of the 23 chromosomes in the secondary spermatocytes is still duplicated, composed of two identical sister chromatids, joined at a central point. The second act of meiosis is about to begin, a swifter, simpler division designed to separate these sisters. The two secondary spermatocytes, without any further delay or DNA replication, immediately enter meiosis 2. This second division is mechanically very similar to mitosis. The 23 duplicated chromosomes in each cell align at the center. New spindle fibers form and attach to the sister chromatids of each chromosome. Again, you can sense the gentle, purposeful tension as the fibers prepare to pull. This time, the goal is to separate the identical copies of each chromosome. The fibers contract, and the sister chromatids are pulled apart, migrating to opposite poles of the cell. Each chromatid is now considered a full-fledged chromosome in its own right. The secondary spermatocyte divides once more, and the result is 4 small circular cells called spermatids. Each spermatid contains 23 single chromosomes, the perfect haploid number required for fertilization. From one initial primary spermatocyte, 4 potential sperm have been created. These spermatids are genetically unique, each one a novel combination of the parental DNA, a testament to the creative power of crossing over. They are small, simple, and round, bearing little resemblance to the streamlined swimmers they are destined to become. But they carry within their compact nuclei the most precious cargo imaginable, a complete, viable, and unique blueprint for a new human life. They are the seeds of potential, the culmination of the intricate genetic divisions of meiosis. These newly formed spermatids are still embedded in the loving embrace of the Sertoli cells, clustered together near the central lumen of the seminiferous tubule. They are connected to each other by thin cytoplasmic bridges, remnants of their incomplete cell divisions. This connection allows them to share gene products and develop synchronously, a small band of brothers preparing for their final transformation together. The Sertoli cells continue to nourish them, absorbing their excess cytoplasm and providing the factors they will need for the dramatic physical changes to come. The journey through meiosis is complete, a passage from a diploid state of 46 chromosomes to a haploid state of 23. It is a process of reduction and recombination, a masterpiece of cellular engineering that ensures both genetic continuity and infinite variety. The quiet, warm environment of the test has nurtured this entire process. The constant supply of testosterone fueling the divisions, the follicle stimulating hormone orchestrating the support system. The slow wave-like progression of development along the tubule ensures that as this cohort of spermatids finishes meiosis, another group of primary spermatocytes is just beginning the journey. The production line is continuous and efficient, a testament to the profound biological imperative to reproduce. The genetic material is now perfectly packaged, halved and shuffled, ready for the next and perhaps most visually dramatic stage of all, the physical sculpting of the spermatid into a spermatozoon. The cell is no longer dividing, but it is about to undergo a radical metamorphosis. Imagine the sheer volume of this activity. Within the intricate network of your seminiferous tubules, this process of meiosis is happening constantly, in countless locations simultaneously. Billions of primary spermatocytes are continually entering meiosis, one shuffling their genetic decks and dividing to create a steady stream of secondary spermatocytes. These, in turn, are rapidly dividing into spermatids. The microscopic landscape is a blur of chromosomal movement, of cellular division and transformation, a silent, ceaseless ballet of creation. It is a process of immense complexity, yet it unfolds with a quiet, reliable rhythm, a fundamental beat in the symphony of life. The genetic potential has been secured, now the physical form must be forged. The spermatids, now nestled in the apical cytoplasm of the Sertoli cells near the tubule's central lumen, are poised on the brink of a spectacular transformation. They have completed their genetic mission, but in their current round, simple form, they are completely unequipped for the arduous journey that lies ahead. They possess no means of propulsion, no way to penetrate an egg, and are burdened with unnecessary cellular baggage. The next stage, known as spermiogenesis, is not a process of division, but one of intricate and profound remodeling. It is the metamorphosis of a simple circular cell into one of the most specialized and streamlined cells in the entire body. The spermatozoon. This is cellular artistry, a sculpting process of breathtaking efficiency and purpose. You can sense this as a shift in the energy within the tubule, from the internal nuclear focus of meiosis to an external structural reconstruction. The first major event is the formation of the acrosome. Imagine the Golgi apparatus, a cellular organelle responsible for packaging proteins, beginning to produce and secrete small enzyme-filled vesicles. These vesicles coalesce and migrate to one pole of the spermatid's nucleus, drawn to what will become the front of the sperm. They flatten and fuse, forming a single cap-like structure that drapes over the anterior half of the nucleus. This is the acrosome, a specialized locum packed with powerful hydrolytic enzymes, such as hyaluronidase and acrosome. These enzymes are the key that will one day allow the sperm to digest its way through the protective outer layers of the egg. The acrosome is a warhead of biochemical agents, assembled and sheathed, waiting for its moment of activation. Simultaneously, as the acrosome caps the nucleus itself begins to change. It elongates, becoming more oval and streamlined. The DNA within undergoes a remarkable process of hypercondensation. The usual protein scaffolding that keeps DNA organized, the histones, are systematically replaced by smaller, more basic proteins called protamins. This allows the genetic material to be packaged in an incredibly dense, almost crystalline state. This extreme compaction serves two purposes. It makes the head smaller and more hydrodynamic for swimming, and it protects the precious genetic cargo from damage during its journey. You can imagine this as compressing a vast library of books into a single, tiny, indestructible crystal, a process of profound physical information density. The genetic blueprint is being locked into a secure, transportable vault. While the head is taking shape, at the opposite pole of the cell, the tail begins to form. The centrioles, organelles involved in cellular division, migrate to the posterior end of the nucleus. One of them docks onto the nuclear envelope and begins to assemble the axon mean, the core of the flagellum. This is a classic 9+2 arrangement of microtubules, a structure conserved throughout evolution for cellular motility, from single-celled organisms to the sperm of mammals. It is a microscopic rotary engine, a flexible whip-like structure that will provide the propulsive force for the sperm's journey. Imagine this filament growing outward, extending away from the cell body, a nascent tail reaching for its future. The cytoplasm of the spermatid begins to shift dramatically. The mitochondria, the powerhouses of the cell, are marshaled with purpose. They migrate towards the growing tail and arrange themselves in a tight helical spiral around the initial portion of the axoneme, just below the head. This section will become the midpiece of the sperm. Think of it as the engine room, a compact, powerful battery pack that will supply the vast amounts of energy in the form of ATP required to whip the long tail and propel the sperm forward. The concentration of these mitochondria is a clear indication of the energetic demands of the journey to come, a testament to the foresight of cellular design. As these structures form, the acrosomal head, the condensed nucleus, the midpiece, and the long tail, the majority of the spermatid cytoplasm becomes superfluous. The cell's goal is to become as lean and efficient as possible. The nurturing Sertoli cell plays a crucial role here. Acting as a cellular sculptor, it begins to absorb the excess cytoplasm from the spermatid, trimming away everything that is not essential for its final function. This shed cytoplasm is then broken down and recycled by the Sertoli cell, a model of biological efficiency. What is left is a lean, mean swimming machine, a head containing the genetic payload and the enzymatic key, a midpiece packed with energy, and a long tail for propulsion. The spermatids remain tethered to the Sertoli cells throughout this entire metamorphosis, which takes approximately 24 days. They are arranged in clusters, their heads buried deep within the protective cytoplasm of the nurse cell, their developing tails extending out into the central lumen of the seminiferous tubule. You can imagine the lumen now, not as an empty channel, but as a field of waving tails, the first sign of the millions of swimmers being prepared for release. The Sertoli cell controls this entire process, providing structural support, biochemical signals, and absorbing waste, guiding the transformation with an unseen intelligence. The cytoplasmic bridges that once connected the brother spermatids now dissolve, and each cell becomes an individual, poised for independence. The level of precision is astounding. The assembly of the acrosome, the compaction of the DNA, the formation of the tail, and the positioning of the mitochondria all happen in a perfectly coordinated sequence. It is a testament to the power of the genetic program encoded within the cell, a blueprint for self-assembly that unfolds with unwavering accuracy. The quiet, warm, and immunologically privileged environment of the adluminal compartment is the perfect workshop for this intricate construction. The slow, forward push from the developing cell layers behind ensures that by the time spermiogenesis is complete, the nearly mature sperm are positioned right at the edge of the lumen, ready for the next step of their journey. The transformation is almost complete. The round, unassuming spermatid has been sculpted into a marvel of biological engineering. It is a cell with a single overriding purpose: to travel, to seek, and to deliver its genetic information. It has been stripped of all non-essential components, its every feature optimized for speed, efficiency, and penetrative power. The final product is a cell unlike any other in the body, a testament to the evolutionary pressures of sexual reproduction. It is a microscopic messenger, a biological arrow, honed and perfected for its solitary mission. The final act within the seminiferous tubule is about to take place, the release of this newly formed spermatozoon into the world. Feel the sense of completion, the quiet satisfaction of a masterpiece finished. Billions of these transformations are happening simultaneously. A continuous process of cellular sculpture, the seminiferous tubule, which began as a nursery for stem cells, has now become a finishing school and a launch pad. The hormonal symphony of testosterone and follicle, stimulating hormone, has guided this process from the very beginning, from the first division of a stem cell to the final elegant sculpting of the spermatozoon. The stage is set for spermiation, the moment of release, the first taste of freedom for a cell born to move. The journey from a simple round cell to a streamlined swimmer is a story of profound change, a visual and structural epic on a microscopic scale. The culmination of spermiogenesis is a moment of quiet release, a cellular letting go. The fully formed, though not yet motile, spermatozoa are now ready to be released from the protective embrace of the Sertoli cells. This process, known as spermiation, is the final act that takes place within the seminiferous tubule. It is not a violent expulsion, but a gentle, coordinated detachment. Imagine the Sertoli cell retracting its final cytoplasmic tethers, like a hand slowly opening to release a delicate creature it has nurtured from birth. The spermatozoa are cast off into the fluid-filled central channel, the lumen, of the tubule. This is their first experience of individuality, their first taste of the world beyond the cellular nursery. With this release, the spermatozoa are now free floating within the lumen, but they are not yet capable of swimming on their own. They are morphologically mature, but functionally immature. Their tails are fully formed, but the biochemical machinery that powers their movement is not yet switched on. They are carried along passively, swept away by the gentle current of testicular fluid that is continuously secreted by the Sertoli cells. You can sense this as a slow, inexorable drift, a microscopic river carrying its precious cargo towards the next stage of the journey. The slow, rhythmic contractions of the myoid cells surrounding the tubule provide a gentle peristaltic push, aiding this passive transport. The fluid in the lumen is a rich, specialized medium, a sort of amniotic fluid for the developing sperm. It is high in potassium, androgen-binding protein, and other factors secreted by the Sertoli cells, creating a protective environment for the newly released spermatozoa. As they drift along the length of the convoluted seminiferous tubule, they are like passengers on a slow-moving river. Their final destination still far ahead. This journey through the remaining length of the tubule is a passive one, a time of transit before their education in motility begins. They are carried towards the central hub of the testis, a network of channels that will collect the output from all the hundreds of tubules. This collection point is called the rete testis, a labyrinthine network of interconnected channels located in the mediastinum testis, the central fibrous core of the organ. Imagine hundreds of small streams converging into a complex web-like delta. The fluid and spermatozoa from the seminiferous tubules all flow into this network, mingling together. The rete testis acts as a collecting and mixing chamber, ensuring a homogeneous mixture of sperm from all the different production sites within the testis. The cells lining the rete testis have a single cilium that beats to help move the sperm along, another subtle mechanism ensuring there is no stagnation in the flow. The journey is always forward, always progressing. From the rete testis, the spermatozoa are propelled into the next set of ducts, the efferent ductules. There are about a dozen or so of these small, delicate tubes, and they form the bridge between the testis proper and the next major organ in the journey, the epididymis. The efferent ductules have a unique lining, a mix of ciliated cells that help sweep the sperm along, and non-ciliated cells that absorb a significant amount of the testicular fluid. This absorption is crucial. It concentrates the sperm, increasing their density in the remaining fluid. You can imagine the river narrowing and deepening, the current becoming denser with its living cargo. This concentration is a key step in preparing the sperm for storage and maturation. The journey through the efferent ductules is still largely passive for the spermatozoa themselves, but it marks a critical transition. They are leaving the environment of the testis, the site of their creation, and entering the long winding road of the epididymis, the site of their maturation. The entire process, from the first division of a spermatogonium to the release of a spermatozoon into the lumen, takes approximately 74 days. It is a long and meticulous process, a testament to the complexity of creating this highly specialized cell. And at every stage, the environment has been perfectly controlled, from the cool temperature of the scrotum to the hormonal milieu within the testis, to the protective embrace of the Sertoli cell. As the sperm enter the epididymis, they are embarking on the next chapter of their existence. The epididymis is a single, tightly coiled tube, so convoluted that if it were unraveled, it would stretch for an astonishing 6 to 7 meters, or about 20 feet. It is here, during their slow passage through this long and winding tube, that the sperm will undergo their final maturation, acquiring the ability to swim and to fertilize an egg. The epididymis is not merely a passive conduit. It is a highly active and sophisticated finishing school for sperm. It has a head, a body, and a tail, and as the sperm travel through these regions, they are exposed to a changing biochemical environment that will trigger their final development. You can sense the change in the landscape. The seminiferous tubules were a vast distributed network of production. The epididymis is a single focused pathway of maturation. All the output of the test is funneled into this one remarkable structure. The journey through the epididymis will take another 10 to 14 days. During this time, the sperm will be profoundly transformed, not in their shape, which is already set, but in their biochemistry and function. The membrane of the sperm head will be modified, proteins will be added and removed, and the internal signaling pathways that control motility will be activated. It is a period of quiet, internal refinement, the final polishing of the key before it is ready to be used. The sheer numbers remain staggering. On this journey, your body is producing sperm at a rate of over 1,000 per second, or more than 100 million per day. Each one of these microscopic cells has undergone the incredible journey we have just witnessed. The mitotic divisions of the spermatogonia, the genetic recombination and reduction of meiosis, the dramatic physical sculpting of spermiogenesis, and the gentle release of spermiation. And now this continuous massive stream of cells is being funneled into the epididymis, a silent, flowing river of immense potential. The story of their creation is nearing its end, but the story of their purpose is just beginning. The journey from the lumen of the seminiferous tubule to the head of the epididymis is a passage from birth to adolescence. The spermatozoa are fully formed but still naive and incapable. They have been created, protected and transported, and now they are delivered to the place where they will learn to become functional. The quiet warmth of the scrotum continues to provide the ideal environment, and the intricate network of blood vessels continues to supply the necessary oxygen and nutrients to fuel this next stage. The body's wisdom is evident in this separation of creation and maturation, allowing each process to occur in a specialized environment, optimized for its unique and delicate tasks. The river of life flows on, carrying the promise of the future on its silent microscopic current. The journey now enters the intricate winding canals of the epididymis. Imagine this structure not as a simple tube, but as a long, serene and highly sophisticated river, its banks lined with specialized cells that actively shape the destiny of the spermatozoa flowing through it. The sperm arrive at the head of the epididymis, the caput epididymid, still largely passive travelers carried by the fluid current. Here the process of maturation begins in earnest, a subtle but profound transformation that will awaken the latent potential within each cell. The environment within the epididymal duct is vastly different from that of the testis, and it is this new biochemical landscape that drives the sperm's final development. As the dense suspension of spermatozoa begins its slow, week-long passage, the cells of the epididymal lining are hard at work. These principal cells, with their long branching microvilli called stereocilia, are constantly modifying the fluid surrounding the sperm. They secrete a complex cocktail of proteins, enzymes, and other molecules, including glycoproteins that will coat the surface of the sperm head. This coating is crucial. It alters the membrane's electrical charge and fluidity, and it masks some of the recognition sites that will later be needed for binding to the egg. This is a process of stabilization and protection, preventing a premature activation of the sperm's fertilizing capacity. It is like placing a safety cap on the tip of an arrow, a cap that will only be removed at the final moments before it reaches its target. Simultaneously, the principal cells are reabsorbing fluid and phagocytosing any abnormal or degenerating sperm, a rigorous quality control check that ensures only the most viable candidates continue on the journey. You can sense this as a process of purification and concentration, a refinement of the sperm population. The fluid becomes thicker, the concentration of sperm higher. Within this evolving medium, the sperm themselves are undergoing deep internal changes. One of the most significant events is the acquisition of forward motility. While the tail, the flagellum, was fully constructed in the testis, the sperm arriving in the epididymis can only manage a weak, uncoordinated twitching. They lack the ability to swim in a straight line. As they travel from the head, through the body corpus, and towards the tail chorda of the epididymis, they begin to acquire this crucial skill. This is driven by changes in intracellular pH and calcium ion concentrations, as well as the phosphorylation of key proteins in the axone and midpiece. A molecule called cyclic MP, CAMP, plays a vital role, acting as an internal messenger that activates the molecular motors within the tail. You can imagine this as the engine being turned on for the first time, the complex machinery of the flagellum being calibrated and synchronized. The weak, erratic movements gradually transform into strong, purposeful, whip-like beats capable of propelling the sperm forward with impressive speed and direction. By the time they reach the tail of the epididymis, the majority of sperm are fully motile, ready to swim. Another subtle but critical change occurs. A small leftover droplet of cytoplasm, which remained attached to the neck of the sperm after spermiogenesis, migrates down the tail and is eventually shed. This is the final streamlining of the cell, the shedding of the last remnant of its immature past. The sperm is now leaner, its power-to-weight ratio optimized for the long swim ahead. It is a process of shedding the unnecessary, of becoming purely and singularly focused on its ultimate function. The journey through the epididymis is a rite of passage, a transformation from a morphologically complete cell to a functionally competent one. The tail of the epididymis, the chorda epididymidis, serves as the primary storage reservoir for these mature, motile sperm. Here, in this cooler, slightly acidic and low oxygen environment, their metabolic rate is suppressed. They are kept in a state of suspended animation, quiescent but fertile, their energy reserves conserved. Imagine a vast, silent army, millions of swimmers held in readiness, their powerful tails still, their energy stores full, waiting for the signal to be deployed. They can be stored here for several weeks, remaining viable and ready for ejaculation. This storage capacity is what allows for the release of a large number of sperm in a single event, dramatically increasing the chances of fertilization. The entire epididymis is wrapped in smooth muscle, which, like the myoid cells of the tubules, contracts in slow rhythmic waves to gently propel the sperm forward along its 6-meter length. During sexual arousal, these contractions will become much more powerful and frequent, especially in the tail and the subsequent duct, the vas deferens, to propel the sperm onwards. But for now, in this state of quiet readiness, the movement is gentle, just enough to keep the population slowly migrating towards the storage depot in the chorda. The sensation is one of profound stored potential, a quiet power held in reserve. The journey through the epididymis represents the final crucial phase of education for the sperm. It entered as an inert, incapable cell, and will emerge as a self-propelled, targeted missile, equipped with the motility and the biochemical stability needed for its mission. The structure of the epididymis is perfectly suited for this task. Its immense length provides the necessary time for maturation, and its specialized lining cells create the dynamic chemical environment that drives the transformation. It is another example of the body's incredible efficiency and design, a dedicated organ for a single vital purpose, the finishing of a sperm. Feel the quiet stillness of the chorda epididymidis. It is a place of waiting, a silent barracks filled with millions of potential lives. The journey from the brain's first hormonal whisper to this point has been long and complex. It has involved intricate hormonal cascades, precise cellular divisions, breathtaking genetic recombination, and profound physical metamorphosis. Every step has been guided by an ancient encoded wisdom, a biological program perfected over countless generations. The sperm are now complete. They are mature, motile, and stable, their genetic cargo secured within their hypercondensed nuclei. The next stage of the journey will take them out of this storage depot, and into the final ducts that lead to the outside world. They will be mixed with fluids from other glands that will provide them with nutrients and a medium for transport. But here in the cool, quiet tail of the epididymis, they wait. They are the culmination of a process of almost unimaginable complexity and scale. Each one is a testament to the power of human anatomy to create, nurture, and perfect. The silence of this waiting is filled with a sense of immense possibility, the latent energy of millions of swimmers ready for the race of a lifetime. The story of their creation is complete. The story of their journey is about to enter its final propulsive chapter. From the quiet reservoir of the chorda epididymitis, the journey of the sperm enters its final conduit before meeting the fluids that will form semen. This next passage is through a long muscular tube known as the vas deferens, or ductus deferens. Imagine a transition from the winding leisurely river of the epididymis to a straight, powerful canal designed for rapid transport. The vas deferens is a thick walled tube, about 45 centimeters long, that ascends from the scrotum through the inguinal canal and arches up, over, and behind the bladder. Its primary role is not maturation, but propulsion. It is the main highway for sperm transport during the process of ejaculation. The wall of the vas deferens is impressively muscular, containing 3 distinct layers of smooth muscle: an inner longitudinal layer, a middle circular layer, and an outer longitudinal layer. You can sense this as a structure of immense strength and potential energy. In its resting state, this tube is quiescent, the mature sperm remaining stored in its initial section, the ampulla, as well as in the tail of the epididymis. But upon the appropriate neurological signal, this thick muscular wall will engage in powerful, rapid, and rhythmic peristaltic contractions. This is the force that will propel the sperm from their storage depot all the way to the ejaculatory duct with incredible speed. It is a biological launch system designed for forceful and efficient expulsion. As the vas deferens loops behind the bladder, it widens to form a section called the ampulla. Here, it is joined by the duct of a crucial accessory gland, the seminal vesicle. The seminal vesicles are a pair of coiled sac-like glands that lie on the posterior wall of the bladder. They do not store sperm, as their name might misleadingly suggest. Instead, their critical function is to produce and secrete the majority of the fluid that will make up the semen. Imagine these glands as bustling chemical factories, working to create a rich, nourishing, and protective medium for the sperm's journey outside the body. As the sperm are propelled through the ampulla of the vas deferens, they are mixed for the first time with this seminal fluid. The fluid from the seminal vesicles constitutes about 60 to 70% of the total volume of semen. It is a thick, alkaline, and yellowish fluid, and its composition is perfectly tailored to support the sperm. It is rich in fructose, a simple sugar that serves as the primary energy source for the sperm's powerful tail movements once they become active in the female reproductive tract. This is the fuel for their journey, the provisions packed for their long and arduous swim. Without this fructose, the mitochondria in the sperm's midpiece would quickly exhaust their limited internal reserves. You can imagine this fluid as a life raft, providing not just transport, but also essential supplies. The seminal fluid also contains prostaglandins, a group of lipid compounds that have a profound effect on the female reproductive tract. Upon deposition in the vagina, these prostaglandins are thought to stimulate smooth muscle contractions in the uterus and fallopian tubes, creating gentle reverse peristaltic waves. This action helps to draw the sperm upward, actively assisting them on their journey towards the egg. It is a remarkable example of biochemical communication, where the semen itself helps to prepare the pathway for its cellular passengers. It is a chemical key that helps to unlock the path forward. Furthermore, the seminal fluid contains a protein called semenogalin, which gives the semen a sticky gelatinous consistency immediately after ejaculation. This coagulating property is thought to help the semen adhere to the cervix, preventing it from immediately draining out of the vagina and giving the sperm time to begin their journey upwards. The alkaline nature of the fluid is also critically important. The environment of the male urethra and the female vagina is acidic, which is hostile to sperm and can immobilize them. The alkalinity of the seminal fluid helps to neutralize this acidity, protecting the sperm and promoting their motility and viability. It is a protective cloak against a harsh new world. The mixing of the sperm from the vas deferens with the fluid from the seminal vesicle marks the formation of the ejaculatory duct. This is a very short tube, only about 2 centimeters long, formed by the union of the ductus deferens and the duct of the seminal vesicle. It passes through the substance of the next major accessory gland, the prostate, and opens into the urethra. The journey is nearing its final common pathway. The sperm are no longer just sperm. They are now a component of a complex, life-sustaining fluid, semen. They are suspended in a medium designed to power, protect, and assist them. The prostate gland itself is the next contributor to this mixture. It is a single donut-shaped gland about the size of a walnut that sits at the base of the bladder, encircling the first part of the urethra. As the ejaculatory ducts pass through the prostate, the gland secretes its own fluid into the stream. This prostatic fluid is thin, milky, and slightly acidic, and it makes up about 30% of the semen volume. It contains a different suite of important molecules. You can imagine another stream joining the river, adding its own unique properties to the final mixture. Prostatic fluid contains citrate, another nutrient source for the sperm. More importantly, it is rich in enzymes that will play a crucial role after ejaculation. One of these is prostate-specific antigen PSA, an enzyme that, after a period of about 5 to 20 minutes, will break down the coagulum formed by the semenogalin from the seminal vesicles. This liquefaction of the semen frees the sperm, allowing them to begin swimming actively out of the seminal plasma and into the cervical mucus. It is a timed release mechanism, a 2-stage process of initial adherence followed by liberation. The prostate also secretes seminal plasmin, an antibiotic protein that may help to destroy bacteria in the male urethra and female reproductive tract, reducing the risk of infection for both partners. This adds another layer of protection to the seminal fluid. The contributions of the seminal vesicles and the prostate gland are absolutely essential for fertility. A man can produce perfectly healthy sperm in his testes, but without the nourishing, protective, and activating fluids from these accessory glands, the sperm would have virtually no chance of ever reaching and fertilizing an egg. The final, smallest contribution to the seminal fluid comes from the bulbourethral glands, also known as Cowper's glands. These are 2 pea-sized glands, located at the base of the penis. In response to sexual arousal, well before ejaculation occurs, they secrete a thick, clear alkaline mucus that drains into the urethra. This fluid serves 2 purposes. First, it lubricates the tip of the penis in preparation for intercourse. Second, and more importantly, it neutralizes any traces of acidic urine that might be left in the urethra, which could be lethal to the sperm. It is a preparatory fluid, cleaning and paving the way for the main event, ensuring the pathway is safe and clear for the semen to pass through. The sperm, now suspended in the rich, life-sustaining mixture of fluids from the seminal vesicles and the prostate gland, have reached the final segment of their journey within the male body. They are in the prostatic urethra, the portion of the tube that passes through the center of the prostate gland. From here, the path merges into the membranous urethra, and finally the spongy urethra, which runs the entire length of the penis. This canal, which normally serves the urinary system, has been prepared for this reproductive function. The neutralizing, lubricating fluid from the bulbourethral glands has already passed through, ensuring the environment is no longer acidic and is ready to receive the precious cargo of semen. The moment of ejaculation is a powerful, reflexive, and highly coordinated neurological event. It is a spinal reflex triggered when the level of sexual stimulation reaches a critical threshold. The process involves 2 distinct phases, emission and expulsion. You can sense this as a gathering storm of neural activity, a culmination of signals that will trigger a cascade of muscular contractions. The brain sends a powerful volley of sympathetic nerve impulses down the spinal cord, targeting the smooth muscle of the reproductive ducts and accessory glands. This is the point of no return. The first phase, emission, is the process we have been building towards. It is the movement of the sperm and seminal fluids into the urethra. The thick muscular walls of the vas deferens begin to contract in powerful, rapid waves of peristalsis, propelling the stored sperm from the epididymis and ampulla forward into the ejaculatory ducts. Simultaneously, The smooth muscle in the seminal vesicles and the prostate gland contracts, squeezing their secretions into the stream. All these components, the sperm, the fructose-rich fluid, the coagulating proteins, the liquefying enzymes, are mixed together in the prostatic urethra, forming the complete substance of semen. At this same moment, a sphincter at the base of the bladder contracts tightly, sealing off the entrance to the bladder to ensure that the semen is propelled forward and that urine does not mix with it. The second phase, expulsion, is the forceful ejection of the semen from the urethra. This is driven by the rhythmic contractions of the bulbar spongiosus muscle, a powerful skeletal muscle located at the base of the penis. These contractions, occurring at intervals of approximately 0.8 seconds, create the pulsatile expulsion of semen from the external urethral orifice. The sensation is one of powerful rhythmic release, the culmination of the entire creative and transport process. An average ejaculation will release between 2 to 5 milliliters of semen, a volume that contains anywhere from 100 million to over 500 million individual sperm cells. This incredible number is a testament to the sheer productive capacity of the test and the evolutionary strategy of overwhelming the odds. Each of these 500 million sperm is a unique genetic entity, the product of the meticulous process of meiosis and recombination you witnessed earlier. Each one carries a perfectly packaged haploid set of 23 chromosomes, a unique blueprint for a new individual. Each one has undergone the dramatic physical transformation from a round cell into a streamlined swimmer. Each one has been matured and empowered in the long, winding school of the epididymis, and now launched into the world, their individual and collective journey truly begins. They have been created, nurtured, stored, and now, finally, deployed. Upon entering the female reproductive tract, the sperm face an immediate series of immense challenges. The vaginal environment, despite the buffering effect of the seminal fluid, remains acidic and is patrolled by the female immune system, which recognizes the sperm as foreign invaders. The thick cervical mucus, which guards the entrance to the uterus, acts as a formidable barrier, a microscopic filter that only the strongest and most motile sperm can penetrate. Millions will perish in these first few minutes, their journey ending almost as soon as it began. It is the first great filter in the race for life. Those that successfully navigate the cervix enter the vast open space of the uterus. Here, they must swim against a gentle downward current of uterine fluid. Their newly activated tails, powered by the fructose from the seminal fluid, now whip furiously, propelling them forward. The prostaglandins in the semen may be gently assisting them, causing subtle uterine contractions that help to pull them upward towards their ultimate destination, the fallopian tubes. It is within one of these two tubes that an egg, if recently ovulated, will be waiting. The sperm do not know which tube holds the prize, so they must divide their forces, with roughly half swimming towards the left tube and half towards the right. This splits the army of survivors once again. The journey up the fallopian tube is perhaps the most arduous leg of the journey. It is a long and complex channel, and the sperm must swim against the gentle downward beat of cilia that line the tube, which are normally tasked with sweeping the egg towards the uterus. Only the most robust swimmers can make this upstream journey. Out of the hundreds of millions of sperm that began the race, only a few thousand will successfully reach the upper portion of the fallopian tubes. And of these elite few, only a few hundred will arrive in the immediate vicinity of the egg. The journey, which can take anywhere from 30 minutes to several hours, is a brutal process of elimination, ensuring that only the fittest and most vigorous sperm have a chance to fertilize. As these final few hundred sperm approach the egg, they must undergo one final crucial transformation, a process known as capacitation. This is a final conditioning that the sperm must undergo within the female reproductive tract. The glycoprotein coat that was applied in the epididymis is stripped away, and the sperm membrane becomes more fluid and permeable to calcium ions. This process makes the sperm hyperactive, causing their tails to beat with even greater force and their heads to swing more widely. This hyperactivated motility is thought to be essential for detaching from the wall of the fallopian tube and for generating the force needed to penetrate the outer layers of the egg. Capacitation is the final arming of the missile, the last step in preparing the sperm for its ultimate purpose. The egg is not a passive target. It is encased in two protective layers. The outer layer is the corona radiata, a layer of follicular cells still attached to the egg after ovulation. The inner layer is the zona pellucida, a thick, transparent glycoprotein shell. The capacitated sperm, upon reaching the egg, must first burrow their way through the corona radiata. Then they encounter the zona pellucida. This is the moment for which the acrosome was created. The binding of the sperm head to a specific receptor on the zona pellucida triggers the acrosome reaction. The outer membrane of the sperm head fuses with the membrane of the acrosome, creating pores that release the powerful digestive enzymes stored within. Hyaluronidase and acrosome go to work, digesting a path through the tough zona pellucida, allowing the sperm to bore its way through this final barrier. It is a moment of intense, focused biochemical effort, the key finally being used to unlock the last door. It is a race against time, as many sperm may be attempting this simultaneously, but only one will succeed. The moment of fusion is the culmination of this entire epic journey, as one single determined sperm successfully digests its way through the zona pellucida. Its head makes contact with the plasma membrane of the egg itself. The two membranes, one from the sperm and one from the oocyte, fuse together, becoming one. It is a moment of profound cellular union, the merging of two separate entities into a single new beginning. The contents of the sperm head, primarily its hypercondensed nucleus containing the 23 paternal chromosomes, are drawn into the cytoplasm of the egg. The journey of the sperm cell is over. Its mission is complete. The instant this fusion occurs, the egg's membrane undergoes a rapid and dramatic change in its electrical potential. This fast block to polyspermy is a nearly instantaneous electrical depolarization that prevents any other sperm from being able to fuse with the membrane. It is a cellular door slamming shut, ensuring that only one sperm's genetic material enters the egg. This is absolutely critical, as fertilization by more than one sperm, a condition known as polyspermy, would result in a non-viable embryo with too many chromosomes. The body's wisdom is evident even in this split-second electrical event. This initial electrical block is quickly followed by a more permanent chemical one, the slow block to polyspermy. The fusion of the sperm triggers a wave of calcium ions to be released from storage within the egg's cytoplasm. This calcium wave propagates across the egg and stimulates thousands of tiny vesicles called cortical granules, located just beneath the egg's plasma membrane, to release their contents into the space between the membrane and the zona pellucida. These enzymes modify the structure of the zona pellucida, causing it to harden and pushing it away from the egg's surface. They also destroy the sperm receptors on its surface, making it impenetrable to any other sperm that may have been boring through. This cortical reaction creates a permanent, impenetrable fertilization envelope, a definitive barrier ensuring the sanctity of the newly formed zygote. With the door now firmly and permanently barred against all other contenders, the egg, which was arrested in the middle of its own second meiotic division, is now spurred to complete it. The surge of calcium that triggered the cortical reaction also reawakens the egg's cell cycle machinery. It quickly completes meiosis II, dividing off a second small polar body, which contains the last of its excess genetic material. What remains is the female pronucleus, containing a perfect haploid set of 23 maternal chromosomes. The stage is now set for the final act of fertilization. Within the cytoplasm of the now activated egg, the sperm's nucleus undergoes a remarkable transformation. The protamines that kept its DNA in a state of hypercondensation are replaced by histones provided by the egg. The tightly packed crystalline DNA begins to decondense, to unfurl and swell, forming a structure identical in appearance to the egg's own nucleus. This is the male pronucleus. For a brief period, these two separate nuclei, one containing the genetic heritage of the father and one containing the genetic heritage of the mother, lie side by side within the vast, nutrient-rich cytoplasm of the single cell. You can sense this as a moment of quiet, profound significance. Two separate genetic histories, two lineages stretching back countless generations, are now brought together within the microscopic confines of a single cell membrane. It is the fusion of two worlds, the creation of a completely novel genetic combination that has never existed before and will never exist again. The potential for a new, unique human life is now fully present, held within this single totipotent cell. It is a moment of pure, unadulterated potential. The two pronuclei, each containing 23 chromosomes, slowly migrate towards the center of the cell. Their nuclear envelopes break down, and their chromosomes, one set from the father and one from the mother, mingle together on a newly formed mitotic spindle. They align at the cell's equator, just as they would in any normal cell preparing for mitosis. This mingling of chromosomes, this creation of a single diploid set of 46, marks the true moment of conception. The resulting cell is now known as a zygote, the first cell of a new human being. It contains all the genetic information, all the instructions needed to develop into a complex organism of trillions of cells. This zygote, this single cell, will almost immediately begin the process of cleavage, the first of many mitotic divisions. It will divide into 2 cells, then 4, then 8, then 16, all while being slowly swept down the fallopian tube by the gentle beat of the cilia towards the uterus. This journey will take several days, and by the time it reaches the uterine cavity, it will be a small, compact ball of cells called a morula. This will then develop into a blastocyst, a hollow ball of cells with an inner cell mass that is destined to become the embryo, and an outer layer that will form the placenta. This blastocyst will then hatch from the hardened zona pellucida and implant into the soft, receptive lining of the uterine wall, the endometrium, which has been preparing for this moment under the influence of hormones. The journey from the initial hormonal pulse in the hypothalamus to this moment of implantation is a story of breathtaking complexity, precision, and synchrony. It involves the creation of a specialized motile cell designed for a single purpose, its journey through 2 different bodies, and its final successful fusion with an egg. The odds against any single sperm succeeding are astronomical, yet the process is so robust and produces such vast numbers that it is remarkably successful. The quiet, constant production of sperm in the seminiferous tubules, a process often occurring unnoticed, is the foundation of all human continuity. It is a narrative that unfolds on a scale from the macroscopic, the entire human body and its systems, down to the molecular, the interaction of single proteins, and the shuffling of DNA. It is a story of communication, of hormones acting as messengers, of cells responding to chemical cues, and of 2 gametes finding each other in a vast and challenging landscape. The quiet warmth of the testes, the cool storage in the epididymis, the powerful surge of ejaculation, the perilous swim through the female tract, and the final dramatic fusion with the egg are all chapters in this incredible biological saga. The legacy of that single sperm, one of hundreds of millions, now resides in the nucleus of every cell of the developing embryo. Its journey is complete, but its information, its unique contribution to the genetic lottery, will be replicated trillions of times over. The whisper of life that began in the deep, quiet center of the brain has now become a shout, a new life embarking on its own journey of growth and development. The microscopic dance of creation has given way to the macroscopic miracle of a new human life, a continuous, unbroken chain stretching back to the very dawn of our species. The journey concludes not with an end, but with a continuation. The implantation of the blastocyst into the uterine wall marks the beginning of a new biological narrative, that of pregnancy and embryonic development. The genetic information so meticulously packaged and delivered by the sperm is now actively directing the growth of a new organism. The hormonal signals that once governed the creation of sperm now shift to support the developing pregnancy, a testament to the body's ability to adapt its powerful chemical language to new purposes. The continuous production of sperm in the testes, however, does not cease. It is a ceaseless underlying rhythm of the male body, a constant state of readiness and potential that persists, day after day, year after year. Imagine once more the seminiferous tubules, the vast 700-meter network of microscopic factories. Even now, as one of their creations has fulfilled its ultimate purpose, the production line continues unabated. New generations of spermatogonial stem cells are dividing. New cohorts of primary spermatocytes are embarking on the intricate genetic ballet of meiosis, shuffling their parental DNA to create novel combinations. Billions of spermatids are undergoing the profound metamorphosis of spermiogenesis, being sculpted into streamlined swimmers. The Sertoli cells continue their tireless work as guardians and nurturers, and the Leydig cells continue to bathe the entire system in the essential fuel of testosterone. The process is a testament to life's relentless drive to perpetuate itself. With every beat of your heart, another 1,000 sperm are released into the lumen of the tubules, beginning their passive drift towards the epididymis. They are a silent, flowing river of potential, each cell a microscopic vessel carrying a unique genetic message. The sheer scale of this continuous production is a quiet marvel. It is a biological insurance policy on a massive scale, a strategy that ensures that despite the immense odds and the perilous journey, the possibility of creating new life is always present. The body invests an enormous amount of energy and resources into this constant background process of creation. The journey through the epididymis is also a continuous procession. A constant stream of newly formed but immature sperm enters the head of this long winding duct, and a constant stream of mature motile sperm is being stored in the tail, ready for the next journey. The epididymis acts as a living, dynamic interface between creation and function, a place of quiet transformation where potential is converted into capability. The sophisticated biochemical environment of this tube, with its secretions and absorptions, continues to provide the final crucial lessons for the developing sperm. It is a finishing school that is never out of session. The accessory glands, the seminal vesicles, the prostate, the bulbourethral glands, also remain in a state of constant readiness. They are continuously producing their vital fluids, their chemical factories humming along, ensuring that whenever sperm are called upon to make their journey, they will be equipped with the necessary fuel, protection, and assistance. The entire system, from the brain's hormonal control center down to the smallest secretory gland, exists in a state of harmonious and perpetual preparation. It is a beautiful, self-regulating system, finely tuned for a single overarching purpose. Reflect on the journey of a single cell. It began as part of a seemingly immortal stem cell population, nestled at the base of a tubule wall. It was selected and committed to a path of development. It underwent the profound genetic divisions of meiosis, becoming a unique haploid entity. It was radically reshaped and streamlined, stripped of all non-essentials, and equipped for speed and penetration. It was matured and educated in a long and winding canal. It was launched as one of hundreds of millions on a perilous mission. It survived brutal filters and navigated a vast and complex terrain, and against all odds, it succeeded. This success story is written into the DNA of every cell of the resulting offspring. The legacy of that one sperm is profound and lasting, but the story of its brothers, the hundreds of millions that did not succeed, is not one of failure. Their collective presence was essential. They formed the wave that was necessary to carry the one survivor forward. They helped to buffer the harsh environment, to overwhelm the defenses, and to ensure that at least one of their number would reach the destination. It is a story of both individual achievement and collective, sacrificial effort. The entire process unfolds with a quiet, autonomous wisdom. It does not require conscious thought or effort. It is a deep, ancient rhythm of the body, a fundamental aspect of its being. You can sense it not as a series of disconnected mechanical events, but as a single, flowing, integrated process. It is a dance of molecules, a cascade of signals, and a transformation of cells, all woven together into a seamless narrative of creation. The feeling is one of wonder at the intricate, nested levels of complexity, from the hormonal symphony orchestrated by the brain to the molecular motors that power the flagellar tail. The promise of new life, which was the driving force of this entire journey, now rests in a different location, the warm, nurturing environment of the uterus. But the potential for creating that promise again remains, undiminished, within the testes. The cycle continues. The hormonal clock continues to tick. The cells continue to divide and transform. The river of life continues to flow. It is a process that connects every generation to the next, a physical cellular bridge across time. It is a fundamental expression of what it means to be alive. There is no final conclusion to this story, because the process itself never truly concludes. It is an ongoing, open-ended narrative of potential. The quiet, rhythmic pulse of creation continues within, a constant reminder of the body's incredible power to generate the seeds of the future. The gentle warmth of the testicular tissue, the subtle flow of fluids, the silent, ceaseless division of cells. All these sensory impressions point to a process that is not a historical event, but a living, breathing, and enduring reality. The wonder is not just in the destination, but in the journey itself, a journey that is happening, right now, within the quiet, protected spaces of your own anatomy. The potential held within each microscopic sperm cell is immense. It is the potential for a new consciousness, a new personality, a new set of dreams and experiences. It is the continuation of a family, a community, a species, and it all begins here, in this quiet, meticulous, and profoundly beautiful process of anatomical creation. The story does not end, it simply pauses, leaving us with a sense of awe at the intricate, self-sustaining, and life-affirming journey that is constantly, silently, and beautifully unfolding within.